Hello, I'm Lisa Tucker-Kellogg from the Duke NUS Medical School, and my co-author is Kang Hyuk Jun from the Department of Biomedical Engineering at the National University of Singapore. Our poster is entitled, Modeling the Indecision Between Inflammation and Proliferation in Chronic Wounds. Healthy wound healing exhibits an orderly succession of three phases: inflammation, proliferation, and resolution. In contrast, non-healing wounds or chronic wounds fail at proliferation, and there are competing hypotheses to explain why. Some of the most widely accepted culprits are bacterial infection, excessive inflammation, and insufficient matrix. Here's our model. Most computational models of wound healing have focused on either the inflammatory phase or the proliferative phase. But in this work, we're developing a dynamical system model that represents the transitions between the three phases as if it's a self-organizing system of mutual regulation. The variables of our model represent known regulators of healthy and chronic wounds: bacteria, immune cells, cytokines, fibroblasts, collagen matrix. The variables that are more apparent to observation in a laboratory environment, the so-called readout variables, appear in blue, and the underlying cell state variables appear in gold. When our model is simulated, it produces a sequence of three phases, each dominated by high levels of one cell state variable: inflammation, proliferation, and resolution. And then we identify some sensitive parameters of the model that can, when we adjust them, change the behavior of the system, change the trajectory so that the whole system enters a non-healing oscillation, also called a limit cycle. For example, when we decrease the parameter for the rate of bacterial clearance, the model gives a steep, fast spike in immune cell activity, and then an earlier transition to fibroblast activity on this axis. This is a phase diagram. And then at the point in time when the system ramps up this proliferation, the bacteria are not yet fully eliminated. That allows the bacteria to regrow during the beginning of the proliferation phase. The regrowth of bacteria causes reactivation of the immune cell activity, and that reverses the accomplishments of the proliferative phase. After this immune cell activity, the system returns back to proliferation, again without clearing all of the bacteria, and so the cycle repeats. So in summary, the way oscillation occurs in this model is to exploit the capacity of a normal wound to move in reverse to adjust in case a wound gets re-injured and reinfected. Another thing we do in this model is to show that when the wound healing system is trapped in a limit cycle, the cell state variables oscillate with significant magnitude, but the observable readout variables have almost undetectable levels of oscillation. So in other words, the wound oscillation would appear static when it's assessed using the available variables because these are time-integrated variables. This prediction is consistent with the fact that chronic wounds don't look like they're obviously oscillating. So in summary, we propose a novel dynamical system in which normal and non-healing wounds both arise from identical influences with only slight differences in the kinetic parameters. We offer an explanation for why chronic wounds appear to have halted healing, and our model combines together multiple previously hypothesized culprits so that we can show why they can all be correct. And in our self-organizing system, each component of the system uses its influence to alter the phase transitions so that the wound can respond to unpredictable threats from the external environment. This ability to respond to unpredictable external threats can then lead to inflammation, disorganization, which is how the wound transitions from a sequential trajectory to an oscillating infinite loop. So that's our poster. Thank you very much. Please have a look at the PDF version so you can zoom it to suit your own eyes. Thanks very much.